There you go. So, actually, I can explain. What was happening is that there are two video cards on my computer. And the video card 1 is controlling this, and the video card 2 is controlling this. So the output are different. And I finally found how to do so. I apologize for the problem. So, now it's very nice and big format screen. So we saw this one, and that's more or less where we were last time, about what the cells are made of. And this section is small molecules in cells, and mostly a cell is made of carbon compounds. And cells contain 4 major families of small organic molecules. They are families of organic molecules. Okay? So this is the building blocks. Okay? Small molecules that make large macromolecules. Okay? So for example, sugars, they will make polysaccharides, glycogen, and starch. Starch in plants, glycogen is in animals. So polysaccharides, glycogen, and starch, they are made of sugars. Okay? Fatty acids, they are the building blocks of fats and membrane lipids. Amino acids, they are the building blocks of proteins, and nucleotides, they are the building blocks of nucleic acids. And we will see in brief what are the characteristics of each of these molecules and macromolecules. First of all, if you actually lyse a bacteria, you break up a bacteria and you see what it's made of, you can see that in percent of total cell weight, 70% of water is more or less like us. Inorganic ion, only 1%. Sugars and precursor, 1%. Amino acids and precursor, 0.4. Nucleotides and precursors, 0.4. So, quite a low amount of these small molecules, right? But if you take a look at the macromolecules, so big macromolecules, then it's 24% of the total mass. So this is just like us. We are made not only of small molecules, but mainly we are made of these big, huge macromolecules like proteins, DNA, RNA. This is really what is a characteristic of living tissues, really. And the second column here, this is the approximate number of different families of these. So for example, water, of course, this is only water. But inorganic ions, maybe a total of 20 types of inorganic ions. Sugars and precursors, 250. It's approximately, okay? But if you check macromolecules, there are a lot of macromolecules, a lot of different things, okay? A lot of different families. So not only in their amount, but also in their complexity, the macromolecules is really mainly what we are made of. By the way, nucleotides and precursors. Can you sort of tell me, can you think of the precursors? Nucleotides, you may already know the structure of nucleotide. But what are the precursors of nucleotides? What are the precursors of amino acids? You know, it's written amino acids and precursors, nucleotides and precursors. What does it mean, precursor? For amino acid and nucleotide. For nucleotides, for example, if you have the nucleotide structure in your head, maybe you don't have already. This is what we are going to see with me, but you don't have. What is a precursor of nucleotide? No? No? From maybe school biology? No? No? Okay, that's alright. We will see that later. But first, let's start with the most simple one, is the sugars. Sugars are very important because they are one of the main energy source for our cells, but they're also the subunits of the polysaccharides, which can have many different functions. And this is the typical structure of a sugar. You can see this is a heterocyclic compound. Heterocyclic because it's composed of carbon and oxygen. And you have usually one hydroxide that is linked to each of the carbon. Not always, but very often. So this is composed of 1, 2, 3, 4, 5, 6 carbon. Okay. And it has one oxygen here. And this is how the molecule looks like. And this is the space filling model. So it's a rather simple molecule. What is this? Do you guess what is this if you count the number of atoms? So C6, H1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, H12, O1, 2, 3, 4, 5, 6, O6, C6, H12, O6, which is also known as glucose. Eh?
A beta group.
Yeah, glucose. Okay. C6H12O6 is glucose. Okay. And but yeah, it can be beta or alpha. Let's not go into too much details today. All right. So this is just a sugar. But they can make polysaccharides by combining with each other. So here we have two monosaccharides. So this only means one unit. And in a condensation reaction, we get rid of one molecule of water. So OH and H, they complex together and they are expelled. And then there is a formation of a glycosidic bond between two subunits. And we have here a disaccharide. Okay. So this is not really a polysaccharide. Well, it's a polysaccharide, but usually in the mind of scientists, when we say polysaccharide is more than two. If you have only two, we say disaccharide. Okay. Then fatty acid chains are components of cell membranes. So the next molecule that we see is a fatty acid. And fatty acid, they are composed of mainly two parts. There is a head, which is usually a carboxylic acid head, and because of its characteristics, it's hydrophilic. Hydrophilic means it likes water, okay? And it has a tail. The length of the tail can be very different, okay? And the shape of the tail can be very different. But here you see it's very simple, CH2, 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 and only at the end, of course, it's CH3. This is a saturated fatty acid. It's saturated because all the available bonds are filled up with hydrogen, okay? And this is a very straight molecule that you can see here in the space filling model. It's a very straight molecule. But please keep in mind it has a hydrophilic head and it has a hydrophobic tail. This is very important for later. But sometimes this fatty acid can make a bit more complex molecule. And for example, here you have one molecule of glycerol that is linked to 3 saturated fatty acid. This is called triacylglycerol, and it's the main storage of fat in your liver. When your liver stores fat, it's mostly under this shape, when 3 fatty acids are complexed to glycerol. And in this case, these fatty acids, they are saturated, and in this case, the fatty acids are unsaturated. unsaturated. Okay? Do you guess what it means? Why do you have a kink here if it's unsaturated? If it's saturated, it's completely straight, as you can see here. And I told you, when it is saturated, all the extra, covalent bonds are used up by hydrogen. But if it's unsaturated, then some of the carbon will have double covalent bond between each other. Okay? That's why there is a kink in the chain. Okay? That's simply why there is a kink in the chain. Because in here, there is a double bond. And because there is a double bond, this causes a change in the angle of the chain. So this is simply because of that. But it looks very simple, but the properties between a saturated fatty acid and an unsaturated fatty acid are pretty different. This is what the cell membrane is mostly made of. You have still a glycerol, but this glycerol is only linked to two fatty acid chain. The available bond is, the last available bond is not with a fatty acid, but with a phosphate group that is itself linked to another polar group. It can be many things. The result is very similar. So you have a hydrophilic head and you have a hydrophobic tail. Okay, even if you have two fatty acids, it's still hydrophobic tail. And each of these molecules, they will assemble with each other to form something that is called a phospholipid bilayer. It's called the phospholipid bilayer because you have a phosphate and it's a lipid, so it's a phospholipid. And it's called a bilayer because you have one layer here and you have one layer there. Can you explain to me why there is two layers? Can you explain with your own words why there are two layers? Note here that it's in water. Okay. If you put this molecule into water, you mix up a little bit. They will form these bilayers. Why? Why these phospholipids will form bilayers? They can form not only bilayers, but they can form also this kind of structure where here this is all the hydrophilic head and the hydrophobic tail are here. And it forms almost like a cell, essentially. That's just like a cell. But why? Why do they form this structure? I want you to explain. Not only to know if it's important, but how to explain it also important. Especially why bilayer? Why not one single layer?
Because phospholipids if phospholipids form single layer, then hydrophobic tails touches water.
Yeah.
So hydrophilic head touches water and hydrophobic tail touches together.
Yeah. Yes, exactly. It's because the affinity of the hydrophobic tail for hydrophobic tail is actually much higher than the affinity with water. So this is why the hydrophobic tail will associate with each other. And the heads will be oriented so that they associate with water molecules, which are also polar. So this is why. And this very simple property is one of the key of why cells are cells. First, if you think about a cell, it's just an endogenous medium surrounded by a bilayer. Okay? Essentially, at the very basic level. And then you can see that, as I told you, some of these fatty acid chain, they can be straight or they can be kinked if they are unsaturated. What do you think what will be the consequences of that? Imagine a bilayer that is entirely made of phospholipids which are composed of saturated fatty acids. Or a bilayer that is composed of phospholipids exclusively made with unsaturated fatty acids. What will be the difference? So the question is, what if you have this? So this is the case where all of the fatty acids are saturated, and the case that you have on the picture here. So you have bilayer, but they are all of different. They are all composed of unsaturated fatty acids. What will be the difference between these two bilayers?
If bilayer is composed of only saturated fatty acid, intermolecular force will be stronger and tight layer, stiff layer is.
Exactly. So this, because all the molecules are very straight, this will be a very stiff bilayer. But here, these unsaturated fatty acids will cause a lot of disturbance in the arrangement. So these bilayers will be much more fluid than this one. And this is actually very important because the bilayer of cells is very fluid. It has to be fluid. It's always in movement because protein needs to be transported and such and things like this. So the composition of these bilayers is very important. Now we are with the amino acids, and they are the subunits of proteins. Fatty acid is very simple. You have a central alpha carbon that is linked to a hydrogen, that is linked to an amino group, and that is linked to a carboxyl group. And it has a very important side chain that is different for each amino acid. Okay? It's called amino acid because this is the amino group and this is an acid. Okay? So in the aqueous environment, they are charged. They are usually in their ionized form. So we have NH3 here and we have COO minus there. Okay, this is the nature in water. So it's very, very small molecule, but they link to each other to form protein. And we usually say that proteins are, there is a polarity in proteins because they are composed of amino acids that are linked into a very specific order. This is called the N-terminus because the last chemical group is the amino terminus. And this is the C-terminus because the last group of the last amino acid is a carboxyl group. Okay. So here you have a small polypeptide that is composed of phenylalanine, serine, glutamate, and lysine. And you can see each of these amino acids, 1, 2, 3, 4, they have different side chain. But they are all bound in the same way with each other. This is very important. And of course, since each of these amino acids has a different side chain, this will give this amino acid a very different property. You can imagine that the phenylalanine here, the property of the phenylalanine will be very different from the property of serine, okay, because it has completely different structure and completely different molecular characteristics. Okay, now we are going to see the nucleotides, which are the components of the nucleic acid. And nucleotides are a little bit more complicated. They are composed of a central ribose, or ribose, that is linked on one side to 1, 2, or 3 phosphate. In this case, it's 3 phosphate. Each of these phosphates is linked to the previous one by what is called the phosphoanhydride bond. And if it has triphosphate, then this is why you say ATP, for example, adenosine triphosphate. And on the other end of the ribose, there is here adenine, which is the nitrogenous base. Okay? It's called the nitrogenous base because it contains a lot of nitrogen. Okay? And it's called the base because it is chemically a base. So that's what is a nucleotide. Okay? That is what a nucleotide is. And nucleotides are different because they contain different nitrogenous bases. They can also be transformed and recycled. So here this is the case of ATP, and there is a phosphoanhydride bond here that is very important between the second and the third phosphate. That can be broken. Okay, it can be broken. Here this is ATP who is transformed into ADP with the incorporation of one molecule of water. And an inorganic phosphate is released in the environment. This bond is very important because when you break it, it provides a lot of energy to the cell. That the energy that the cell can then use for all sorts of stuff. Okay. Most of the things that you do when you think, when you run, what you are doing, essentially, you are breaking this bond of all the ATP you have in your cell. This gives you a lot of energy. Okay, so please keep that in mind. The ATP is very important to provide energy. But then, when you have used a lot of ATP, for example, after your baseball match, then you need to replenish your ATP, and you can do that by actually, again, adding a new phosphate onto ADP to regenerate ATP. Okay. And different cells regenerate ATP in different ways. For example, a plant may just regenerate ATP using sunlight, which we cannot do. Okay. When we want to replenish ATP, we have to burn something else, like glucose or fatty acid. All right? And here you have an oligonucleotide, where also here there is a polarity. This is the 5' end, which is a convention. The 5' end is this phosphate end. And then we have the first nucleotide here that is linked to a second nucleotide that is linked to a third, to a fourth. And the last available group here is the OH, which is called the 3' end. So remember, there is a polarity in protein, and there is also a polarity in oligonucleotides like this one. And in this case, they are all different. All these nucleotides are different because they have different nitrogenous base. Here we have guanine, adenine, thymine, and cytosine. And you can see they are all different. Please keep that in mind. Alright, now we have seen very small building blocks, and we have seen that they can, they can bind to each other, and they can form slightly bigger assemblage. But here we are going to see what if they start making really huge macromolecules. Each macromolecule contains a specific sequence of subunits. So again, let's imagine a very simple organism, like again a bacteria, because it's very easy to imagine. It's made, as I said, 70% of oxygen and 30% of chemicals. But most of these chemicals, as we saw before, is made of macromolecules. Macromolecules are DNA, RNA, protein, and polysaccharide. Protein is really the main fraction. This is true for bacteria. This is also true for our own cells. And as I said before, this is just a cartoon. Sugar, when they form polymer, it's called a polysaccharide. A polymer of amino acid is called a protein. And a polymer of nucleotide, it's called nucleic acid. And an important reaction is the one we have already glimpsed at. It's a reaction of condensation, where one molecule of hydrogen is released and a new link is made between two subunits. This is very general. It's just like these squares, because it can be anything. This is the very similar reaction for most of the polymerization of organic molecules. So for example, here you imagine, okay, I will come back later to this. This is the second part. Okay, so I'll go to this a little bit later. Kind of, should I explain now? I'm a little bit confused. What should I explain now? Yeah, okay, let's do it now. So, of course, now we know what proteins are. The sequence of protein very often is known for most of the protein that make up your cell. But it was not always known like this. And scientists were interested to know whether these huge molecules are mostly made of covalent bonds between each of the subunits, or if it's not all covalent bonds. Maybe they were thinking that what we have is a protein, but that is composed of many small subunits that are all linked together not only with covalent bond, but also with non-covalent bonds. So this is the two hypotheses are here. In case the protein that you are studying is only made up of covalent bond, then all the amino acids are linked together. But you could imagine a protein that exactly has the same composition, but in which some of the amino acids are linked covalently, but some of these amino acids are linked by non-covalent bonds, like hydrogen bonds. So what you want to know is which is true. Is this one true, or is this one true? And the question is how do we know? And it's still now, it's the same thing. If you are studying your macromolecules, how can you know if the macromolecules is a mix of different things that are all linked together by a non-covalent bond, or it is just a huge molecule, one single molecule? How could you know? What sort of techniques could you use to know, okay, this is true, or this is true? What could you do? What experiment could you do for that? This is especially true for proteins.
X-ray.
X-ray, yes, yes. More simply, yes, centrifuge. Yes. But before that, you can centrifuge, engines loop. Centrifuge, you could centrifuge. But the problem, if you take a protein, especially a big protein, or, I mean, even though they are, in this case, they are non-covalent bonds, and only one covalent bond may break very easily, but there are a lot of non-covalent bonds. And so many that actually, if you centrifuge, A or B may end up going at the bottom of your tube without difference. So before centrifugation, you should do something to make sure that what you have is one single molecule, or what you have is a polymeric protein. So centrifuge is a very key. This is the final step. What could you do? There are different ways you could achieve that, but mostly one that is probably easier. When the protein is in its native form, it's very big and it's unyieldy, and you may not really know what it's made of because it's just too big and too, the amount of interactions that the protein does with itself is just too much. You must first do something to analyze the protein, to centrifuge. What could you do that would help make a difference between A and B? Think about the relative strength between a covalent bond and a non-covalent bond. So for example here, let's say, and that's the case, mainly these will be hydrogen bonds. So think about the strength. What is the strongest in terms of energy? Is it a hydrogen bond or is it a covalent bond? The strongest, it's a covalent bond, right? The strongest. So then what could you do?
Heat.
Heat, yeah. Heat, it's an idea. Heat, it's an idea. Heat will do something to the protein. Heat will denature the protein. But very often it's just, it may make things worse because the protein will all sort of congeal together. Just like, for example, imagine you take an egg and you heat the egg and it becomes all stiff and white. So that's one way to do things, but it may not be the best. Although it's a good idea. Actually, in addition to heat, you may also do something else. This is what I always do in the lab here when I want to analyze protein. Yes, I heat, but before heating, I must do something.
Acid.
Use acid. That's interesting. That's an interesting proposal. Also, you need to be careful because if the acid is too strong, it may break a lot of stuff. But yeah, you use some chemicals. The idea is very good. You need to add some chemicals. What kind of chemicals could you use? The idea is good, right? You have to use chemicals that will break some of these things. It's actually quite very frequent chemical. We probably use it every day. Water. course, yeah, you need to have water. But you need something else, because these proteins will be like this in water. Okay? So you need something else. I can think of two chemicals that are quite good for this purpose. One you may not know, the other one I think you know, but you don't think about it. Simply soap, detergent. Detergent. If you add detergent to it, it will break up all these non-covalent bonds will be broken up, okay? And so what you want to do is to break up these non-covalent bonds, and then, as you said, use centrifuge. By the way, centrifugation is just taking a sample, and this is the mix of protein that you have, and you have a specific substance. It's called here a sucrose gradient, which means that it's composed of water plus sucrose, but the concentration of sucrose is different. High up there, it's not so concentrated, and at the bottom, it's quite concentrated. And then you centrifuge, so it means that you make this spin very, very quickly. You can change the speed. And since there is a centrifugal acceleration that is created, if you have a heavy protein, it will go down very quickly. If it's very light, it will stay up in the tube for quite a long time. The retention will be much longer than a very heavy protein. And what you have, if the mix that you have is composed of very different things, of a lot of different stuff, of different size, what you have is a completely heterogeneous aggregate. But if you have one single substance, one single molecule, then what you have is one very specific band here. For example, imagine what you put here is a single solution of hemoglobin. It's one single protein. Then it will actually sediment as a single band. Now, this is the idea. Okay, so if you do this, what would you have for this case? And what would you have for this case? The one at the top here and the one at the bottom corresponds to which one? That's easy. If you denature this and you centrifuge, what do you obtain with A? Do you obtain this or do you obtain this? And what do you obtain with B? Do you obtain this or do you obtain this? That's easy. Donnez des moyens. Don't be afraid. Which is which?
A single band.
A is, of course, a single band. And in this case, you have a mix of different size molecules. So each of these molecules will sediment at a different, so you have a smear of the exact. All right. So in our cells, most of the polymer are, of course, under a stable conformation. But you can devise conditions that will disrupt these non-covalent bonds, and this is what we were speaking earlier. Although it is very easy to disrupt covalent bond, I told you, you can add heat, you can add detergent, another molecule, and another agent to disrupt non-covalent bond. But some proteins are very, very tightly bound to each other, and this non-covalent bond, as we saw before, is very important not only for proteins to associate with each other, but for specificity. For example, imagine a macromolecule A, for example, a protein, and it has a very specific shape. Okay, the molecular shape is very specific. And this molecule A encounters 3 different macromolecules, like 3 different subunits, different proteins. And A and B, they can make some of the non-covalent bonds with each other, but somehow it's a very poor match. A and C is a little bit better. It's a little bit more tightly associated, but still it's very easy to dissociate again. So they will associate. This will happen. But the problem is that they will not stay associated. So they will spontaneously revert to their condition. They will associate, they will separate. They will associate, they will separate. Okay. But if A encounters its partner, then the association is such that it becomes very stable. And the time they will spend associated is much longer than the time they will stay non-associated. So this becomes the nature of specificity. When 2 molecules are just fit with each other, they associate with each other with such a stability that they will stay most of the time together. And this is only because of the shape of the protein and the non-covalent bond they can make between each other. So you can imagine the situation where you have different subunits, so you have a mix of amino acids and nucleotides. When you create covalent bonds, you make, for example, RNA molecules as well as protein. And now, if you put non-covalent bond into the equation, not only you have RNA molecule and globular protein, but you have something completely different. And that's, in this case, a ribosome. Ribosome, do you know what is ribosome? What is the function of ribosome? What is the function of ribosome? What do they do?
Protein synthesis factory.
Yes. It's a factory. It's really, the word is nice. It's a factory. Scientists like to say this is a molecular machine. It's really huge. It's 30 nanometer in diameter. It's a really, really huge complex. You can actually see this ribosome in the electron microscope because it's so big. And it's composed of RNA molecules and proteins, but only because of non-covalent bonds. So even though I told you non-covalent bonds, they are not very tight, they are not very strong. If you make many of them, you can give a seemingly complete random mix of RNA and proteins into a molecular machine that has a very specific function, only due to a very tight association between all the subunits because of non-covalent bonds. All right, I will skip this guy. So you can take a look at the book. This just tells you that carbon can, each atom of carbon can make different links with other carbons in different ways. You can have chains, you can have branch trees, you can have rings. As we say, rings, as we saw, rings is very important, for example, for nucleotides and sugar as well. And covalent bond, I will not see that. I think this is known. Okay, it should be known. So you have CH compounds that we have seen in fatty acids. If you have a very simple compound, this is methane, CH4. And if you have CH3, try to remember this one, it's a methyl group. CH3 is a methyl group. So here, for example, that's a methyl. And carbon can make single bonds with each other, like in this case, but it can also make double bonds or even triple bonds. This also exists. You have CO compounds. So I will not go into detail with this. Okay, it's in the book. You can look at it by yourself. I will not ask you what it is. Okay, this will not be a part of the exam. It's out of the scope of this course, I think. And it's not that interesting. You can look. Right, phosphate, we saw phosphate. Hydrogen bond, so I will also skip this one. We already saw what they were, okay, hydrogen bonds. And this will come into this one here. So what we have seen is that, so that's a molecule of water, and of course, each molecule of water can make hydrogen bonds with its neighbors. Okay, this is very basic. And that's why water is liquid at room temperature, because of all these associations between each molecule. But water is also very important because it will determine what kind of associations molecules that are in water will do. So for example, if you put a sodium ion in water, what happens is that all the water molecules will associate with this sodium ion in a very specific way, where you have the partial negative charge of the oxygen will interact with the plus charge of the ion. If you have chloride, that's the same thing, but in the opposite ways. It's negatively charged, so you have the partial positive charge of all the hydrogen that will actually associate with each other. So that's very, very important. And that's why sodium chloride, that's why you can dissolve it in water quite a lot, because the affinity between each of the ions and water is quite high, all right? But then if you have polar substance like urea, this is urea, CONH, you can see that there is a lot of available atoms for hydrogen bonds. So you have a delta plus charge here that will associate with the delta negative charge of the oxygen, and so on and so forth. So all these molecules that can make a lot of hydrogen bonds with water will be very easy to dissolve in water. But if you have hydrophobic molecule, like for example what we saw, these fatty acids, they have no possibility to do hydrogen bonds. And what will happen is that all the molecule of water will be repelled, and they will form a cage around the hydrophobic molecule. So the hydrophobic molecule will be isolated. Okay? So this is why this is isolated from water, so water around it will be highly organized, will form a cage, and this is why when you have a lot of these hydrophobic molecules, they will be pushed together by water. And this is why if you have a lot of these things, they will tend together, just like these bilayer do. Water as solvent, this is very basic. Water as solvent, if you put, for example, a sugar crystal into water, and then each single molecule of glucose will dissolve into water. And each of these single molecules will make a lot of hydrogen bonds with water, and this is the phenomenon of dissolution. All right, acids. That's not interesting. Okay, so let's start from this one here, which is interesting because it gives you more insight on the structure of these fatty acids. So we saw earlier that fatty acids can be unsaturated, they can be saturated, and the length of the hydrophobic chain can be different. So what you have here, it's a C18 chain, this is stearic acid, and here this is only C16 chain, this is palmitic acid, it's two different fatty acids. But in this case, although the representation is not exactly correct, this is a chain that has here one double bond between two carbons. And because of this, it will not be a straight line. So you can take a look here. So this is oleic acid. It's the same as this one. But while each of the saturated bonds will always be straight, because of this, you have a kink in the fatty acid. Okay, just because of this double bond. Triacylglycerol, what we saw before, but now we saw a bit more precisely at the molecular level how it looks. So you have 3 fatty acids that are bound to this glycerol here, which is a very simple molecule. Glycerol is, you can see here, without any other molecule attached to it. Okay. Alright, so this is the same thing. We saw the phospholipid before, but only at the very general level. Here you have the molecular level of this phospholipid. So you have 2 fatty acids linked to a glycerol that is itself linked to a phosphate. And the phosphate is linked to another molecule we see, which is also sometimes hydrophilic. In this case, it's a choline. It can be different things. We will see that. We will see that later. So, I think maybe it's important to make a difference between 2 different things. So, a normal lipid, a phospholipid, has a hydrophobic tail and a hydrophilic head. So when you mix them with water, they can form something that is called a micelle, like this, or they can form a surface film, right? But this is a little bit different from phospholipid. Here you can see this is one single layer, and the center is completely hydrophobic. There is no water inside. When you get phospholipid and glycolipid, it's a little bit similar, they actually form a bilayer. And that is very key because the center is very different from this center. The center here will be filled with water, just like a cell is, right? So please remember this. So, lipid is just something that, it's a very general name. A lipid is just something that is hydrophobic, okay? So, what you have is this one here. This one is a lipid. But you have other lipid, like this is also a lipid. Okay? This is also a lipid because it is hydrophobic. As you can see here, lipids are defined as water-insoluble molecules that are soluble in organic solvents. 2 other common types of lipids are steroids and polyisoprenoids. Both are made from isoprene subunits. So this is isoprene subunit. And if you actually make a polymer of isoprene, what you have is a polyisoprenoid. And you have many repetitions of this isoprene. This is also a lipid, okay? Because it's just something that is hydrophobic. Steroids, so you can see the molecular structure of steroid is completely different. But it is also a lipid, okay? In the mind of some people, they think that this is a lipid. It is true, okay? This is true. But a lipid is more general than that. A lipid is anything that is hydrophobic. Like, for example, what you have here is cholesterol, which also exists in our cell membrane. And you can see here, it's just hydrophobic. There is here a little hydrophilic head here, but mostly this is hydrophobic molecule. Testosterone, which is made from cholesterol. What is testosterone? What is testosterone? It's just written there. It's a male hormone. Okay. Glycolipids, they are just lipids that are linked to a sugar. Okay. Phospholipids, they are lipids, they are fatty acids that are bound to phosphate. A glycolipid is a phospholipid. It's a fatty acid that is linked to a sugar. Okay, that's easy. Alright, so that's... So it's a little bit chemical, but now let's go into the amino acids. As I told you, the amino acids are different because they have different side chains. Okay? And the side chain can either be acidic, basic, uncharged polar, or non-polar. And there are about 20 common amino acids that all your proteins are made of. Right? So the basic side chains, they are, for example, lysine, which is written lys or K. And you can see this is basic because it ends up here with a base. Then you have arginine, which is also basic. In this case, you can see it ends up with a completely different group. Histidine, it's also basic, but the structure is different. Not only the size, but the structure is different. Amino acid, we saw that already. That's very cool, sorry. I know you have that in the book, but I will never ask you from memory to tell me what is the structure of each amino acid. I will not ask you that. It's not really important for this class. Okay. So you can take a look and you can remember that amino acids have different side chains that give them very different properties. But I will never ask you at the exam what is the side chain of asparagine. Okay. That's not important for this lecture. Okay. That's not important. You can take a look, you can remember if you want, but I will never ask you. It's not relevant for this class. No, I want to skip this. Do you think it's interesting? We are all sleeping. I'm also sleeping. Sorry, I had a very late night. Okay, so let's go a little bit. Let's finish this chapter at least. So this is the nucleotides. It's a little short survey on nucleotides. And nucleotides, I told you, 1, 2, 3 phosphates bound to a ribose that is bound to a nitrogenous base. Okay? But you have different nitrogenous base that makes two families of nucleotides. You have pyrimidine nucleotides and you have purine nucleotides. Pyrimidine nucleotides are made of uracil or cytosine or thymine. The purine nucleotides, they can be adenine or guanine. And you can see the structure of the nitrogenous base is different. Pyrimidine, it's only one single heterocyclic cycle. And in the case of the purine, it's a double cycle. Okay, so just please remember these two types of nucleotides. So which associate with which? Which associate with which? This we should all know. So the answer should come like after 1 second. In DNA or RNA, which nucleotide associates with which nucleotide?
Adenine and thymine.
Adenine and thymine.
Guanine and cytosine.
Yes, exactly. Although in DNA, yes. In DNA, A and T, G and C. This is called Watson-Crick base pairing. Watson-Crick base pairing. But in RNA, there can be other non-Watson-Crick association. But you are right, the main one is thymidine together with adenine and guanine together with cytosine. So this is very interesting that one pyrimidine base will associate with a purine base. And one purine base will associate with the other pyrimidine. So you don't have two pyrimidine or two purine together. Okay. Phosphate, as in A and P, so this is adenosine monophosphate. 2 phosphate, this is adenosine diphosphate. 3 phosphate, this is adenosine triphosphate. That's very easy. So again, we saw the same thing. And as you know, maybe you don't know, but in chemistry, each of the carbon is numbered. So this is the 1' position, this is the 2' position, this is the 3' position, the 4' position, and the 5' position. And actually the base also, they have their own number, but let's keep on the details here. Nucleic acid. That's what we have already seen. Okay, let's make a break. Okay, let's take 4 or 5 minutes break. Let's open the window and let's take a break. Ask the worst chapter.
Thank you for uploading slides, one page. But,
Yes.
but I mean, like this, full page, no border, no margin.
Yes, thank you, but I tried, and as I told you, it's too big.
Oh, too big.
It's too big. So I can only upload less than 5 megabytes. And if I make a PDF of the slides at the lowest resolution, it's 10 megabytes.
Oh.
So, but that's okay, right? The one I put, it's big enough, right? Right? The one I just put today.
I just want to reduce paper use. So
Then you can print on double side.
I want to print according to the complexity of slide.
If you want me to pull full size, that means, the way I understand what you mean, is that I have one A4 page, and the slide is there. How is that going to reduce paper? As opposed to have one page and one slide here, which is there. So I don't understand. It's going to be the same number of pages. Really? No?
I can, with this, I can change settings to 4 slides or 1 page or 9 slides in 1 page or 16.
Yeah, but you can also do with that, isn't it?
Too small and
But so the problem is this one. So this is what I have here, you see. This is what I have here. So this is 1 slide per page. But as you can see, this one is 10 megabytes. And I cannot upload on classes. But if you have a USB key, you can take it out. If you want.
yeah.
Yeah, you can. If you have USB key, you are welcome to take it. So I tried, but it did not upload. So this is why I did the one with the one at the center of the page. Any question, by the way? No question?
Like terminology is difficult.
Yeah, sorry, it's very strange today. What terminology?
Like all the names and stuff. Like,
Yeah.
I remember like some familiar from the like what I learned in Finland like in high school, but then most because they're of course in English, it's like
Yeah, I think I would say it's a problem of biology. Like, of course, this chapter was mostly very basic chemistry and stuff like that, but the names of stuff in biology, there are really so many different things, and different names have different connotations. So that's one of the main problems, I think. So, do you have your book here? No, it's quite big to bring. So, about the chapter 1 that we saw first lecture, and then chapter 2. So essentially, what I consider that you should remember that is interesting for this lecture, it's the main pages. So from page 39 to 65. So that's the chapter 2 here. Chemical components of cells. Okay? And after the chapter 2, you have a lot of plates like this, with all sorts of different molecules and different families and things like this. You can take a look if you want, but I will not interrogate you on that. That doesn't tell me whether you are able to think or not. Okay, it's just different shapes of molecules. Okay, just take a look, read, but do not remember. You don't need to remember. So again, all these plates at the end, it's not needed. So, all right. So the previous chapter was very descriptive on the type of molecules and some very simple interactions they can make with each other. But a cell is just more than the sum of its molecules. What is really important is how these molecules interact with each other and why do they interact. Okay, why do they interact? So let's see first of all a few very simple rules about the use of energy by the cells, free energy and catalysis, and activated carriers and biosynthesis. This is essentially a very simple picture of what happens in our cell, where we have molecule A is transformed into molecule B using a catalyzer, which can be enzyme 1. And then molecule B is transformed into molecule C with another enzyme, enzyme 2. The reaction 1 and reaction 2, sometimes they are carried out by the same enzyme. Okay? And all our metabolism, everything that our cells do is just a chain of that. It's a chain of catalyzed reaction. But what are the rules for these reactions to occur? This is what we are going to see in this chapter. It's much more interesting than the previous one. So, we eat food to get energy. Okay? You need to eat glucose, you need to eat fatty acid, you get energy from it, and your body is doing work. During this work, your body will burn some molecules to get energy. and you will have a lot of garbage that you have then to evacuate. So this relationship between anabolic reaction and catabolic reaction is very important. So the food molecules that your cell absorbs, they are broken down via something that is called catabolic pathways. And these catabolic pathways generate useful forms of energy, as well as heat that is just dissipated into the environment. And this also generates many building blocks that are required for then the anabolic pathways to kick in. Anabolic pathways will capture some of this energy and use these building blocks that were released from the catabolic pathways and will synthesize more complex molecules that form the cell. So please remember these catabolic pathways and anabolic pathways. This is quite important. There will always be lost heat. You can see even though these catabolic pathways generate lost heat, it's actually not at least directly taken up by the anabolic pathways. We are not 100% efficient, so some of the energy will always be dissipated as heat. All right. Biological order is made possible by the release of heat energy from cells. This is an example of some of the very complex structure that exists in life. This thing cannot happen spontaneously. You can see it's highly ordered. Okay? So usually what it means is that if it's highly ordered, you need a lot of energy, not only to make it, but to keep it. If you don't give energy to that, it will sort of degrade. Okay? So you must always spend a high amount of energy to maintain this kind of structure. This is a virus. I think this is a section in one of the cilium in the bacteria. This is a pollen grain. This is a section in the stem, I think, and that's a kind of it's a plant. Okay? And you can see a different level of, this is almost at the molecular level here, this is at the cellular level, so you can see all levels are highly organized. And it's very important to keep these things and to spend a lot of energy doing so. If you do not, this is what happens. So the cell is like this very nicely organized room. We all have been kids. And I know myself that when I was a kid, my room was very rarely like this. My room was very rarely so organized with all my books lined up. Everything was probably open on the floor, like this. Okay? And this is exactly the picture that you must have of the cell. The cell is like this. The cell is like, if it's like this, it's dead. Okay? If it's like this, it's dead. But then, when you were kids, and still maybe now, what happens when you have that? And if this stays for quite some time, what will happen? Well, dad or mom will come and say, you know, you have to keep your room, and then you have to make order, and then a lot of energy has to be put in for this to happen. If you don't give energy, this will never by itself, order. It's not a self-assembly. So, that's a spontaneous reaction, okay? As time elapses, this will go like this, and that's an organized effort requiring energy input, which is catabolism, which is anabolism. Which is catabolism, which is anabolism?
Anabolism is energy input.
Yes.
Catabolism is spontaneous reaction.
Exactly, absolutely. Okay, so if you imagine a cell that is in a sea of matter, I like this term, in a sea of matter, so all the atoms in the medium will move and will shock with each other, okay, they will. interact, and so there is a lot of chaos. And inside the cell, all the molecules also have these random motions. Some of them will have shock with each other, and then this will generate some heat. Okay? If the energy increase, the amount of movement and the amount of interactions will also increase, and the heat that is being generated will also increase. However, as you can see this one here, the inside is not so different from the outside. Okay? What is the real case of a cell is like this, where all its components, all its macromolecules are well ordered. And to achieve this, it must generate heat. All these reactions requiring order to be made will generate heat. Because energy will be consumed to create this order, and because energy is consumed, heat will be generated. Okay? So the heat that is generated by the cell will communicate to the environment, and as you can see here, compared to the previous situation, the randomness of the sea of matter, of the water, for example, will increase. So the energy that is burned for the creation of order by the cell is, a sum of it is released as heat, communicates to the environment to increase the disorder. What does it sound like? Do you know of something you may have learned previously? A kind of very important concept in physics. This is an explanation of what happens at the cellular level of this very important rule in physics. So you have a cell who must increase its internal order, and when it achieves this, it generates heat that communicates to the environment and increases the disorder of the environment. So as a whole, if you sort of sum all these order-disorder level, it actually, the whole thing doesn't change much. So what does it sound like? Can anybody tell me?
Energy conservation.
Which is what?
Which is what?
Energy. That's the first one. That's the first one. Energy is not created, is not lost, it is only transformed. That's the first law of thermodynamics. This is the second law of thermodynamics, which is what? Entropy. It's something about very nice. Entropy is a synonym of disorder. Entropy means chaos. Entropy means disorder. And what does the second law tell us? What is the second law? If you think of the second law, then you think, okay, it's not possible for a cell to survive, because the content of our cell is so organized that it seems to contradict the entropy principle. But this tells us that actually no, because this highly organized cell actually has increased the entropy of the whole isolated system that is in there. So what is the second law? Can anybody, do you remember what is the second law? No?
The second law, the second law of thermodynamics is that the entropy can only increase. In an isolated system, the entropy can only increase. Okay? So that's the law. But then, if you think that the cell has a very highly organized, it's very highly organized, so how can it be possible? Because the second law of thermodynamics tells us that the entropy can only increase. So the cell will instantly die. But actually no, because as I said, while the cell is highly organized, all these exchange of matter and energy to make it so organized have actually increased the chaos of the system in which see. Okay, so anyway. Cells can convert energy from one form to another, and this is where the first law comes in, where energy is always transformed. So you have the case where potential energy due to the position actually is transformed into kinetic energy because of the movement of the brick falling down, and when the brick touches down the ground, then heat is released. So this is where potential energy is transformed into heat energy. Or you have the chemical bond energy that is contained in two hydrogen molecules and one oxygen gas molecule, and then you can actually make water. So you have the rapid molecular motion in H2O, which is kinetic energy, and then you have the heat energy that finally is generated because the heat is dispersed in the surrounding. Then you have a third case in which a chemical bond energy, like the one you have in battery, is actually converted into electrical energy, then to kinetic energy. And you have the sunlight that is used by plants that have chlorophyll in their chloroplast. Chlorophyll molecule gets excited and actually can synthesize a lot of complex molecules because of this energy. And this is electromagnetic energy, which is sunlight, that is converted into high energy electrons and then to chemical bond energy. And this is where I will leave you today. So we will go a little bit further in this chapter the next time, and we will see how photosynthesis happens and how the cells get all this energy from oxidization of organic molecules. Okay, so if you have any questions, don't hesitate. So again, please do not remember all chapter 2. All the end of the chapter 2, please do not remember. For this course, it's not needed. Certainly, you must remember probably from chemical course or whatever, but not for this.